Hey, Dr. Stansel, it's Ashley Allen. So today is September 26, 2018. It's for microbiology. This is week 5, our carbohydrate test with fermentation. You can tell I'm a little disappointed. So I obviously don't have my feelings well. I'm a little disappointed in the lab because I keep trying to be very meticulous and I don't know what I'm doing wrong, but I'm not getting results that I think I'm supposed to be getting. And I'll explain further what that is. So basically, the first video, you observed me, break up some of the carbohydrates. We had glucose, fructose, and mannitol. All of those went in 2 vials each. S. cerevisiae and then S. epidermidis, and we're seeing, how they mix together along with the Durham tubes. I got all the air out of those, so I was pretty good at that. That took some technique. And then I allowed these guys to wait. So after 12 hours, what I saw was they were all yellow, maybe a little orange, and at the very top there is a red ring. And I was like, but they're all the same color. I imagine that some of them are going to be a little different. Which one has gas, which one can ferment, which one's a non-fermenter. So I decided to wait a little longer, and according to our lab assist, our instructions, we can wait up to 18 hours. So I waited till 17 hours. With no avail. There's not much of a difference with any of them. The red now is completely gone, so none of them are red. And I'll show you what I have. So just to recap, the carbohydrate test determines the byproduct of fermentation and differentiates among species of bacteria, right? So orange is a fermenter, yellow is a fermenter with gas, and red is a non-fermenter. So remember, we actually started out with red, but we're gonna see if any of our carbohydrates in combination of our broth bacteria would maybe keep it the same, maybe just the acid level would change. But anyway, so let me show you what I have. So here's glucose with S. cerevisiae. You see it's yellow. There's a tiny little bubble in there, not much. Okay, so there's one. Then there is glucose with S. epidermidis. See it? Yellow as well. And then we have fructose with S. epidermidis. Little orange in the tube. I don't know if that means it's because it didn't mix up, or if it's more concentrated in the Durham tube. Here is fructose, S. cerevisiae. See, it's a little orange in there. I'm not sure what that means, to be honest. I would, if you don't mind explaining that to me. And the last two are S. epidermidis with mannitol. Let me get that focus for you. And see, that has a little red in the Durham with a little bubble. And the very last one is S. cerevisiae with mannitol, and that also has, let me bring the tube closer. See how red that one is? So I don't know, again, if it just didn't mix in there and that's why it's still red. That's my conclusion. So technically, all of mine say that they're fermenters with gas. I'm not sure if that's right, but that's my results. Those are my results. I don't know if there's a contamination with one of my broths, and that's why I have an error. I don't know. Help me. But I'll take pictures and upload them and discuss it in my lab review. Thank you.